On July 5, 1996, in a quiet corner of the Roslin Institute near Edinburgh, Scotland, a miracle occurred. But it didn't arrive with a flash of light or a roar of thunder. It arrived with a soft ba and a wet nose. To the naked eye, she looked like any other Finn Dorset lamb. She had white wool, a sturdy frame, and an inquisitive nature. But this lamb was about to shatter our understanding of biology forever. This was Dolly. For the first time in human history, we had done the impossible. We had taken a cell from an adult animal and used it to create an identical twin, born years after its predecessor. Dolly wasn't just a sheep. She was a biological carbon copy. She was the first mammal ever cloned from an adult somatic cell. Welcome to FirstoGraphy. Today, we are diving deep into the genetic dawn, the story of Dolly the sheep, the science that created her, the controversy that followed her, and the legacy she left for all of mankind. Before Dolly, the scientific community believed that once a cell became specialized, like a skin cell, a heart cell, or a brain cell, it was locked. You couldn't turn back the clock. A skin cell was destined to be a skin cell until it died. But to scientists at the Roslin Institute, Ian Wilmut and Keith Campbell had a different theory. They believed they could reprogram a cell, essentially hitting the factory reset button on its DNA. The process they used is called Somatic Cell Nuclear Transfer, SCNT. It sounds like science fiction, but the execution was a masterpiece of patience. Here is how they did it. First, they needed a donor. They took a cell from the mammary gland of a 6-year-old Finn Dorset white sheep. This was the source code. Next came the egg. They took an unfertilized egg from a Scottish blackface sheep and removed its nucleus, effectively stripping away its own genetic instructions to make room for the new ones. Then, the fusion. They placed the adult mammary cell next to the empty egg and used a tiny pulse of electricity to fuse them together. Finally, the spark. That same electric pulse tricked the egg into thinking it had been fertilized. It began to divide, growing into an embryo. This wasn't a test tube baby in the traditional sense. There was no sperm involved. It was a genetic photocopy. We often talk about Dolly as a singular success, but we rarely talk about the 276 times the scientists failed. Creating Dolly wasn't a lucky one-off. It was a grueling marathon of trial and error. Out of 277 attempts at fusing cells, only 29 embryos survived long enough to be implanted into surrogate mothers. And out of those 29, only one resulted in a successful, live birth. Imagine the tension in that barn in July 1996. The researchers had spent years being told that cloning an adult mammal was biologically impossible. They were working in secret, knowing that, if they succeeded, the world would never be the same. When Dolly was finally born, she weighed 6.6 kilograms, about 14.5 pounds. She was healthy, she was strong, and most importantly, she was a Finn Dorset born to a Scottish blackface surrogate. Because she had a white face, while her surrogate mother had a black face, it was visually obvious that she was a clone. The DNA matched the donor perfectly. You might wonder why she was named Dolly. In a somewhat cheeky nod to her origin, Ian Wilmut suggested the name because the donor cell came from a mammary gland. He famously said, "We couldn't think of a more impressive pair of mammary glands than Dolly Partons." But while the name was a joke, the reaction from the public was anything but funny. The birth of Dolly was kept secret for 7 months while the team prepared their scientific papers. When the news finally broke in February 1997, it was like a bombshell. The media went into a frenzy. Within days, the ethics of cloning were being debated in every household. If we could clone a sheep, could we clone a human? Scientists, theologians, and politicians grappled with the God complex. President Bill Clinton immediately called for a ban on federal funding for human cloning research. The Vatican issued a stern warning. Science fiction movies like The Boys from Brazil or Jurassic Park, suddenly felt like documentaries. People were terrified of a future filled with designer humans or clone armies. For the rest of her life, Dolly lived at the Roslin Institute. She was treated like a queen, but she was also under a microscope literally. Scientists watched her every move. Would she age faster? Would she develop clone-specific diseases? Dolly led a relatively normal sheep life. She mated with a Welsh mountain ram named David and eventually gave birth to 6 lambs of her own, Bonnie, then twins Sally and Rosie, and finally triplets Lucy, Darcy, and Cotton. This was a massive scientific milestone in itself. It proved that clones could reproduce naturally. However, the shadow of her origin always loomed. In 2001, at the age of 4, Dolly developed arthritis. People pointed to her cloning as the cause, claiming she was born old because her DNA came from a 6-year-old sheep. Then, in 2003, Dolly began to cough. A CT scan revealed tumors in her lungs. This was a common sheep disease called JSRV, but because of her high-profile status and the risk of her suffering, the team made the heartbreaking decision to euthanize her on February 14, 2003. She was only 6.5 years old, about half the lifespan of a normal Finn Dorset sheep. Was Dolly a mistake, or was she the key to the future? The legacy of Dolly isn't actually about creating copies of animals. It's about cellular reprogramming. Because of Dolly, we learned that cells are plastic. This led directly to the discovery of induced pluripotent stem cells, which later won the Nobel Prize. Today, we use the lessons from Dolly to save endangered species, like the northern white rhino. We use them in medicine, creating animals that can produce life-saving proteins in their milk to treat hemophilia. We are even exploring how to grow human-compatible organs to end the donor waiting list. Dolly now stands in the National Museum of Scotland in Edinburgh. She is preserved for future generations to see, a quiet, woolly reminder of the day humans stepped into a new era of biology. Dolly the sheep was more than an experiment. She was a mirror held up to humanity, forcing us to ask, just because we can do something, does it mean we should? She showed us the fragility of life and the immense power of our own curiosity. She was the first of her kind, but she certainly won't be the last. Thank you for joining us on this journey through time here at FirstoGraphy. What do you think about the ethics of cloning? Should we be cloning extinct animals, or even humans? Let us know in the comments below. Don't forget to like, subscribe, and hit that notification bell to keep exploring the firsts that changed our world. Until next time.